Who works now and stuff like that. So she was just on the phone. Like Ellen? Yeah. Oh my god, great, that's hilarious. Yeah. And then the other one was
These people, they're healthy practices.
and he was like,
Probably hoping next year. Yeah, I mean, Mikhail wants me to
It's always like this.
Yeah.
I've gone live.
I don't sorry.
I just started going live.
Okay, that's
That's kind of what I'm trying to tell.
Yeah. I had that short, like my days of 3
Because he was in the same situation. No regrets if you.
always things that I love. Yeah. You did the presentation. For 1.5 Life is this. You know, and it can make you really.
Hey, David.
Trying to put on the 5
Oh, yeah, we have this knocking construct where we can, where we can knock in a rescue transient to the ABS. So right to cook like the entire sort of TET inducible system, and so you can like clone your gene, and then there's there's an antibiotic resistance, and then P2A, and then RTTA, transcriptional activator.
It is a dot. That's the dot.
But the resistance in the vector is my cell line is already resistant, so I have to switch it out. And yeah, he spent like months cloning this monster. So
Mine is here.
yeah.
I'll take a look at
Okay,
this.
thank you.
What versions we have.
Thanks.
We've made so many, I'm sure.
I was figuring you guys might
None
have
of them.
one.
So we'll expect them to. Is it Valentina there?
Katya? Oh, okay.
Here's Bob.
Oh, really? Oh, okay. It's in life science. Oh, okay. Okay. And what company?
Good.
Okay.
Yes.
Oh, great.
Yeah, that would be great.
It's amazing. Yeah, it's really great. Yeah. Hey, how are you?
Oh, here she is. Yeah, in Nelson. Okay. All right. So now the committee is here. Okay, this is blinding me. So we can start with the official event, and this is a very important day for Gian Paolo Sepulveda, who is a PhD candidate in the program of genetics and genomics in the program in biomedical sciences. It's an important culminating moment for a lot of work, very hard work, brainstorming, worries, writing. So now it's like all coming to the end. And for me, it's a very important day because Gian is my first PhD candidate who is defending his thesis today. He's also very special and kind of new for me that Gian actually did, his model organism was not C. elegans. So this is really first for me and makes it very special also. So as many of you know, Gian started his PhD in 2017 when he was accepted to PIBS program. And it was very lucky for the program. He had great credentials, academic, but also research experience in a program through NIH. And I was thrilled when Gian got interested in my research. And then when he wanted to join my lab, of course. And he's been through different phases of the lab. He was a junior member of the lab and worked with postdocs. For a few years now, he is actually a senior member of the lab, an advisor or mentor to undergrads. There was a period in the middle that was coincided with COVID that was trying period where Gian was my lab, and also that he went through some period of doubt, which happens to graduate students, but he persevered. And I want to say that he's a great colleague, he's a great friend. That's why I see a lot of his friends here. He's wonderful to give advice and help anybody who needs his help. He's excited to present his work, and we traveled internationally, and he's a good travel companion, and we had a good time in Glasgow. But scientifically, Gian really was interested in a project related to cancer. And we have this exciting but very challenging project that had a beginning. A lot of people contributed to the beginning of the project. It was solid, but there was no conclusion. And we really needed to basically overcome this hurdle and find a reasonable conclusion for the story. And Gian was up to the challenge. And I'm very proud of his accomplishments. And especially the last two years where he designed beautiful experiments, and seeing that his hypotheses were right is very satisfying, I'm sure. And he essentially did genetic epistasis analysis in cell culture, like you would do in C. elegans. And speaking about novelty of what he found, if you do search in PubMed and you put dot, MYC, and proteasome, meaning combination of those terms, there is only one result, and this is our bioarchive preprint. And so it's in review in G&D, but it shows you that it was a conclusion for Gian's thesis, but it's actually a beginning. What he's done is a beginning for the field, I think, and the field that is not one field; it's a combination of apparently different fields. And yeah, I'll give you Gian. Won't take more of his time, and I hope you enjoy it.
Can everyone hear me good? Great. Thank you, Alla, for that very kind introduction, and thank you to all of you for making the time today to listen to me talk about the research I've been working on in Alla's lab for the past 6 years, revolving around the non-canonical role of DOT1L in MYC-driven transcription. So, yes, great. All right, so the research I've been working on in Alla's lab for the past 6 years really builds on some literature that challenges the static dogma of transcriptional activators. And what I mean by this is when we think about transcriptional activators, we usually think of a transcription factor that binds to the target promoter of its target gene, and this would lead to expression. Whereas when the transcription factor is not present, it usually leads to low or basal expression of the target gene. However, I hope to show you today a particular example where we have a transcriptional activator binding to the promoter, but this binding is not sufficient to drive the gene expression of its target genes. And I'll show you this through two different stories in this talk, the first of which is how DOT1L mediates the degradation of MYC, leading to target gene expression. And the second is this unusual role of DOT1L for proteolytic activity on MYC. So a little bit of background about MYC, which is probably the most well-known and most well-studied proto-oncogenes of many different cellular functions. So MYC is a transcription factor, and it regulates a lot of the core essential processes of the cell. In particular, it exists in a heterodimer with its binding partner MAX, leading then to the binding at the target promoters called E-box promoters, and this leads also to the recruitment of certain cofactors that would then lead to the expression of target genes, mostly related with cell proliferation and growth, nucleotide metabolism, and ribosome biogenesis. So these are some of the core essential processes of the cell, and as such, MYC needs to be very tightly regulated, especially in non-transformed or normal cells. And so this form of regulation often occurs transcriptionally, as well as through post-translational modifications. And this allows then for the cell to regulate the levels of MYC in order to then regulate gene expression. And when MYC is dysregulated, this can often lead to cancer malignancies. So upregulation of MYC is often found from in around 50 to 70% of cancer malignancies. And I'm showing And I'm showing you here just two separate cancer types, colorectal cancer and head and neck squamous cell carcinoma. And you can see here in the green line for colorectal cancer and here in the red line for head and neck squamous cell carcinoma that patients with higher expression of MYC often correlate with lower survival rates compared to those patients with lower MYC expression. Because of its nature as a transcription factor, MYC is often deemed to be undruggable because of the many intrinsically disordered regions that it has. So it makes it very hard to develop small molecules against the function of MYC. And apart from that, high MYC expression often drives relapse in these patients. So how does MYC drive the expression of its target genes? MYC over here is comprised of many conserved sequences that are called MYC boxes that I've highlighted here in the green boxes over here. And they're highly conserved sequence among MYC paralogs, particularly MYC-L and MYC-N, as well as across different MYC homologs in different species. And I've highlighted here that, that from MYC box 0 to 2, this is often noted as a transactivation domain, which is essential for the expression of MYC target genes. And you can appreciate that these conserved MYC sequences are often binding sites for a lot of the different MYC cofactors. And particularly, we have, I've highlighted here in the red asterisk, histone-acetyltransferases that can bind to MYC, and often this would lead to acetylation of the histones and a more open chromatin context to allow then the expression of MYC target genes. But more importantly, in the transactivation domain, we also have the binding of PTFB, which is part of the super-elongation complex. And this is essential for MYC to promote transcriptional elongation during promoter proximal pausing. So often in early elongation, the RNA often forms this tight complex with the DNA, and the RNA pol2 has tight binding with the general transcription factors and early on at the promoter, which would then lead to the pausing of RNA polymerase to near the promoter. And this pause state is often stabilized by pausing factors like NELF and DSIF. So the role of MYC in this particular case is that it can recruit PTFB, which is a kinase, leading then to the phosphorylation of the C-terminal domain of RNA pol2, as well as the phosphorylation of these pausing factors like NELF and DSIF. This causes certain pause factors like NELF to then fall off of this transcriptional complex, leading then allowing RNA pol2 to have this conformation that allows transcription to restart and to have productive elongation. So there are a lot of factors that can be associated with elongation, but there are also certain chromatin modifications that can be associated with elongation rates, one of which is histone 3 lysine 7-9 methylation. You can see here in these two different studies that genes with higher elongation rates often have higher occurrences of H3K79 dimethylation, especially in the transcriptional start site. In the separate study, when they bin the genes according to their elongation rates, the genes with the highest elongation rates in the red curves over here also have the highest levels of H3K79 dimethylation. So here, we often associate H3K79 with higher elongation rates as well as active transcription. Now, the only enzyme that methylates H3K79 is DOT1L, and it only methylates this lysine in the context of nucleosomes. So DOT1L often has a lot of different binding partners that have been characterized. The most famous of which would be AF9 and AF10. And in this complex, DOT1L is recruited to the target promoters, leading then to the methylation of H3K79 at nucleosomes near the transcriptional start site and the early gene bodies. Now, unlike MYC, which is a well-established oncogene, DOT1L is only now starting to be recognized as an important oncogene in solid cancers, especially in cancers like breast cancers as well as prostate cancers. Here, we can see that increased expression of DOT1L is often associated with lower survival rates. You can see here in triple-negative breast cancers as well as prostate cancers, and these survival curves at the red lines, patients with higher expression of DOT1L often have lower either relapse-free survival or overall survival rates compared to the patients with lower expression of DOT1L. And this connection to cancer doesn't stop there, because H3K79 is often correlated as well with MYC binding genome-wide. So here we're beginning to see this relationship between DOT1L and MYC, and this was solidified in 2015, where they showed that DOT1L and MYC can exist in a particular complex that can then lead to target gene expression. So here, this is an overexpression experiment from 2015, where they overexpressed DOT1L and MYC, and you can see here that MYC co-immunoprecipitates a .1L, and when they do the inverse experiment, .1L co-immunoprecipitates with MYC as well. When they performed siRNA experiments against .1L and looked at the expression of 3 epithelial to mesenchymal transition genes that are also MYC direct targets, you can see that siRNA against .1L leads to the lower expression of these 3 MYC target genes. And in the end, they're able to form this particular model where DOT and MYC can exist in a particular complex, which allows then DOT to be brought to these target promoters to then methylate H3K79 at these target nucleosomes. And this whole process leads to the expression of the MT-related genes. Apart from that, in Ala's lab, we were able to establish the relationship between the homologs of MYC and .1L in C. elegans, in particular MML1, which is a homolog of MYC, and .1.1, which is a homolog of .1L. So we did chip qPCR experiments against MML1 and looked at the MML1 occupancy at the .1 target promoters as listed over here. And we can see here that MML1 does bind to these 4 .1.1 promoters. Now to look at the effects on gene expression, we did a microarray using MML1 mutant worms and compare that to wild type. And here, this is just shown in our volcano plot here. So the genes to the left of the volcano plot are the genes that are downregulated in the MML1 mutant compared to wild type, while the genes on the right are the ones that are upregulated in the MML1 mutant compared to wild type. Now to compare that with the gene expression changes with .1.1, we then did this microarray again with the .1.1 mutant and looked at what happens to gene expression changes here. And we colored this volcano plot accordingly. So the genes that are downregulated in the .1.1 mutant are colored in blue, while the genes that are upregulated in the .1.1 mutant are colored in red. And so here we're really seeing that the gene expression changes between these between MML and .1L and .1N are similar here. So the main question I had during my time at LSAB is really how does .1L and MYC co-regulate gene expression and transcription? And so I'm going to first go to the first part of my talk, which is about how .1L can stimulate MYC activity by promoting its degradation. And to begin to answer this question, we first attempted to develop .1L knockout cells in the triple negative breast cancer cell line where .1L and MYC were first shown to cooperate with one another. And so we used a guide RNA against the first exon of .1L. And you can see here in our second round of CRISPR-Cas9, the .1L protein is decreased. And so we wanted to then get a clonal population of these .1L knockout cells. And we're able to get a .1L haploinsufficient cell line where the first allele has an early stop codon, and the second allele of the cell line has a 15 base pair in frame deletion at the histone methyltransferase domain. So when we looked at one of the main enzymatic functions of .1L, which is H3K79 methylation, we can appreciate here that .1L knockout cells have almost near complete aggregation of H3K79 dimethylation compared to our wild-type control cells. When we looked at cell proliferation, which again is one of the main cell processes that MYC can control, you can see that .1L knockout cells are slower in proliferation compared to our wild-type controls. So we then, with these knockout cells, we performed proteomic and RNA-seq analysis. And over here, and we looked at the upstream regulators that are inhibited in our network. And you can see here that the MYC network seems to be one of the top upstream regulators that are inhibited in our .1L knockout cells compared to our control. The boxes that are colored in blue have a lower activation Z-score, indicating that they are downregulated, while the boxes that are more orange are more activated in our cell type. And so you can see here that MYC is, again, the top regulator. However, other members of the MYC family network, particularly MLX-IPL, as well as the MYC paralog MYC-L, are also inhibited in our proteomic and RNA-seq analysis. When we did gene set enrichment analysis in our RNA-seq data, you can see that a lot of the hallmark MYC targets are downregulated as well. And we confirmed this using some RT-qPCR, using some 8 direct MYC targets. And we can see that the .1L knockouts have downregulation of MYC target genes compared to the wild-type cells. Now, to see if MYC and .1L can interact with one another, we overexpressed flag tag and HA tag .1L into these cells. And you can see that, unsurprisingly, because .1L and MYC both have very apparent nuclear functions, they both colocalize in the nucleus. However, surprisingly, We also see that they often co-localize in these speckles or these distinct puncta when we overexpress them in our cells. More importantly, we did co-immunoprecipitation experiments of MYC and .1L, and you can see here that when we overexpress flag tag MYC and HA tag .1L, we can pull down flag tag MYC over here, and .1L co-immunoprecipitates with MYC. Now, when we do the inverse, where we can pull down flag tag .1L, HA tag MYC can as well be pulled out, indicating that .1L and MYC can interact with one another in a particular complex. So when we think of a transcriptional activator, we often think that this transcription factor binds to the target promoter, leading then to the expression of target genes. And when it is not present, it leads to low or no basal transcription of these target genes. And so now, with our RT-qPCR and RNA-seq data for MYC target genes, we can then assume that MYC binding to the promoter is decreased in our knockouts compared to our wild type. Apart from that, when you look at the MYC protein levels in our knockout compared to wild type, MYC protein levels are slightly decreased in our knockout compared to wild type, and thus, giving us more reason to believe that with lower protein levels, lower expression of MYC target genes, we would then expect that MYC binding to the promoters in our knockout cells would be decreased. However, this is not the case when we looked at MYC occupancy at these target genes. Instead, in our .1L knockout cells, you can see that the occupancy of MYC at the target promoter is often up in these knockout cells compared to the wild type cells. And this leads us to this strange phenomenon that kind of fights with the dogma that we have, wherein we have increased MYC binding to the promoter, but despite this, we have low gene expression. So to recap what I've shown so far, when .1L is present, MYC binds to the promoter at lower levels, but we have higher gene expression of target genes. Whereas when there is no .1L, more promoters are being bound by MYC, however, there's low target gene expression. So the main question then becomes, why does MYC accumulate at the target promoters when .1L is depleted? And why does accumulated MYC not stimulate transcription? And in order to start to answer these particular questions, we have to go back in our dogma of transcriptional activation and transcriptional activators. And in our studies, we found this particular model that ties the ubiquitin proteasome system with transcription factors. In this model, a transcription factor can bind to the target promoter, leading to gene expression. But it becomes a spent transcription factor that needs the help of the ubiquitin proteasome system to then be ubiquitinated, degraded, in order to free up the promoter, then allowing a new transcription factor molecule to bind, leading then to the expression of its target genes. Now, when the ubiquitin proteasome system is inhibited or perturbed in any way, this spent transcription factor remains in the promoter, and occupies the promoter, leading then to the inhibition of transcription of its target genes. So kind of this particular model ties in the degradation cycle of transcription factors and the cycling at the promoter to the expression of its target genes, showing that the degradation is inherent for expression. And this was shown for MYC in a 2016 paper, where they were able to make these cell lines with either the wild-type version of MYC or a lysine-less version of MYC, as shown in the blue bars. And this lysine-less version of MYC is not able to be degraded in the canonical ubiquitin proteasome system because ubiquitin is often deposited to lysine residues. And so in this particular system, when they induce expression of wild-type MYC, this is sufficient to drive the expression of target genes. However, using the degradation-deficient version of MYC, you can see that this is insufficient to drive MYC target gene expression. When they looked at the occupancy of MYC at these target promoters, you can see that the lysine-less version of MYC, that is degradation-deficient, often has a higher accumulation at the target promoters compared to the wild type. Apart from that, the degradation cycle of MYC was linked also in 2003 to its expression of MYC target genes, where they showed that the E3 ligase skip2 degradation of MYC was actually essential for the expression of its target genes. So when they inhibited or depleted skip2, this would often lead to higher MYC protein levels, but despite that, MYC target gene expression was down. So putting these models of degradation cycling and transcription together, we build upon this particular model, wherein MYC can bind to the target promoters, leading then to the expression of MYC target genes. MYC is then ubiquitinated and degraded from chromatin, freeing up the promoter, allowing a new MYC molecule to bind and then lead to transcription. So we wanted then to ask whether .1L depletion affects c-MYC turnover by the proteasome. And so we looked at some of the dot-1L interactors from this 2021 paper, and we see that the proteasome is among the top pathways that are overrepresented in this gene list. So we wanted to ask, does dot-1L depletion affect this ubiquitin proteasome system? And to do that, we made use of this proteasomal inhibitor, MG132. So MG132 would inhibit the degradation of ubiquitinated proteins, and in the case of MYC, it will inhibit the degradation of ubiquitinated MYC. And when we inhibit that degradation, we would expect then to have this accumulation of ubiquitinated proteins. So when we looked at what happens in our dot-1L knockout cells and comparing that to the wild type, you can see that MG132 treatment leads to this accumulation of ubiquitinated proteins. And when you compare just DMSO-treated dot-1L knockout cells in control, you can see that this ubiquitination smear is still present at pretty much similar levels, indicating that dot-1L depletion does not really perturb ubiquitination. Apart from that, when we did MG132 inhibition of the proteasome, we do see the accumulation of MYC over here that we can pull out using the N-terminal antibody of MYC, and we can see that MYC is still being ubiquitinated in our knockout cells and our wild type cells. Now, next we look at the expression of MYC target genes upon proteasomal inhibition, and you can appreciate here that under dot-1L wild type conditions, proteasomal inhibition leads to this trend of a decrease in the expression of MYC target genes. When we looked at what happens in our dot-1L knockout cells, however, this is dampened. This effect is dampened in our knockout cells, indicating that the proteasome and dot-1L seem to be working together in the regulation of MYC target gene expression. In particular, they could be working together in the same pathway or in a similar pathway together. To further establish this connection with the proteasome, we made use of this inhibitor called TAC243. TAC243 is an inhibitor of the E1 ubiquitin activating enzyme. And so what happens when you use TAC243, this inhibits ubiquitination of proteins. So in the case of MYC, it will inhibit the ubiquitination of MYC. However, prior ubiquitinated MYC will still be degraded by the proteasome. And so we did a TAC243 chase experiment to kind of probe at how proteins are being depleted or cleared in these particular cells. In our control wild type cells, we can see here that inhibition of ubiquitination using TAC243, you can see that ubiquitinated proteins are already being cleared as early as 2 hours. And you can already see the effects of MYC accumulating, which we assume to be non-ubiquitinated MYC because we're inhibiting ubiquitination, already starting at 2 hours. Now, when we compare that to the dot-1L knockout cells, however, this effect is significantly delayed, where you see the protein clearance of ubiquitinated proteins later on at 8 hours and not really at 2 hours. And the effect of MYC also only shows up later on at 8 hours, indicating that dot-1L depletion leads to this slower clearance of ubiquitinated proteins. So putting that together in our model, we can see so far that dot-1L and the proteasome are working together for the turnover of MYC at these target promoters, and this turnover is important for the expression of the MYC target genes. Now, when dot-1L or the proteasome are perturbed or not present, this would lead to the accumulation of MYC at the target promoters, but lower expression. We then wanted to ask whether there is a step between ubiquitination and the proteasomal degradation of MYC that dot-1L affects, because there can be multiple steps between that first ubiquitination to the degradation of the protein. And we focus on the valosin-containing protein, VCP, or P97, which has unfoldase and segregase functions, especially in the chromatin. So the VCP exists in a hexamer over here, and it acts after the ubiquitination of its substrate proteins. You can see here that VCP recognizes ubiquitinated substrates, especially substrates that are either, in this example, in a complex with other proteins, or factors that are on chromatin or stuck to a particular membrane. And what it can do is it can extract these proteins from the complex or from the membrane, leading then to the degradation of the substrate via the proteasome, or in some cases, with the ubiquitinases, these substrates can be recycled. So VCP has been shown in multiple compartments to be important, especially for the degradation cycle of proteins. But again, not all proteins, only certain proteins need VCP for their degradation cycle. But more importantly, the ER-associated degradation is one of the most well-characterized functions of VCP, where VCP is needed to extract certain proteins from the ER lumen onto the cytosol, leading them to their degradation. VCP has also been shown to have chromatin-associated degradation, where VCP is important for the extraction of certain DNA damage response proteins, some transcription factors, as well as histones, and this allows the DDR pathway to pursue. Now, the connection between MYC and VCP has been established in 2018, where they use NMS873 to inhibit the function of VCP. So when they use NMS873, you can see that the amount of ubiquitinated proteins increases because, again, we're inhibiting the degradation of a subset of ubiquitinated proteins. When they look at the protein levels of MYC, you can see that NMS873 inhibition leads to the increase in the protein levels of MYC. However, despite that, you can appreciate that in these MYC target genes, depletion of VCP leads to the downregulation of these MYC target genes over here. So again, this goes back to this idea that the degradation of MYC is inherently tied to the expression of its target genes. So they come up with this model in 2018 where VCP is important for the extraction of MYC from chromatin and from its heterodimer binding partner max, leading then to its degradation. And when we put this into our existing model that we're building upon, we can then see that MYC binds the promoter to induce gene expression, and VCP is needed then for the extraction of MYC from the target promoter, leading then to its degradation cycle. So we wanted to ask, does VCP play a role in dopamine-mediated turnover of c-MYC? So in order to begin asking that, we overexpressed dopamine and VCP together and wanted to see whether they interact together in a complex. And you can see here that we can immunoprecipitate dopamine and VCP co-immunoprecipitates with dopamine. We wanted to also see whether dopamine depletion affects VCP levels in any way. However, in our knockout cells, comparing that to our control cells, it doesn't really seem that VCP total VCP protein levels are changed in our knockout compared to our control. And when we detected the hexamer over here using blue native page, you can see that the hexamerization of VCP is also unaffected. So we use NMS873 in our experimentation to inhibit VCP, similar to the 2018 paper. And we can appreciate here that ubiquitinated proteins in both the knockout cells and the control cells also accumulate when we inhibit VCP. And when we look at the protein levels of MYC, we also see the accumulation of MYC protein levels in the knockout and control cells upon NMS873 inhibition. Now, looking at the gene expression of the MYC target genes upon inhibition of VCP, you can appreciate that in these MYC target genes, we have the downregulation of MYC target gene expression. And this is completely abrogated when we inhibit VCP in the dopamine knockout cells. So we also so this basically indicates to us that VCP and dopamine seem to be working together in the same pathway to then regulate MYC target gene expression. We also used another method in order to perturb VCP levels using shRNA. And you can see here that shRNA against VCP would also lead to the accumulation of ubiquitinated proteins. And when we use shRNA against dopamine, we see that H3K79 dimethylation levels are perturbed. When we look at the target gene expression of MYC target genes, you can see here that in the gray and brown bars, SHVCP leads to the downregulation of VCP. And here in the blue bars for dopamine, we can see that SH.1L leads also to the downregulation of .1L mRNA. Now looking at MYC target genes, you can see here that shRNA against VCP leads also to the downregulation of MYC target genes in a similar fashion to what happens when we inhibit or deplete .1L. Now going back to our TAC243 experiments to see how protein clearance is affected in their under VCP and .1L depletion conditions, you can appreciate that under SH control conditions in our triple negative breast cancer cells, we see that again, the clearance of ubiquitinated proteins already starts occurring at around 2 hours. However, this clearance of ubiquitinated proteins is very much delayed in the SH.1L cells. We only see it here at 4 hours, and with the SH-VCP cells, we're only seeing it later on at around 4 to 8 hours later. So this brings us then to our particular model of VCP and .1L cooperation, wherein MYC binds to the target promoter to induce gene expression, and .1L and VCP work together to remove MYC from the target promoter. This leads to the degradation of MYC from chromatin, allowing then this promoter region to be unoccupied, allowing the cycle to continue and induce gene expression. Under .1L depletion conditions, however, MYC still binds to the promoter to induce gene expression. However, removal of MYC by BCP is compromised in this case. Therefore, MYC remains more so at the target promoter and blocks the new transcription factor molecule from binding to induce gene transcription. And this whole cycle is then blocked. Now, for the second part of the story, I'll be talking about proteolytic cleavage of c-MYC by DOT1L, and this shows another layer of regulation of c-MYC by DOT1L. So in our study, during my time in Alice's lab, when we looked at MYC localized to the chromatin, we would often see these shorter MYC fragments, C-terminal MYC fragments, as highlighted over here, and they often show this ladder-like phenotype in our Western blots. Apart from that, when we overexpress HA-tagged and flag-tagged MYC in HEC293 T cells, we would often see that the full-length MYC protein is being produced. However, shorter MYC fragments are also being produced whenever we do the overexpression experiments. And so we thought that these shorter MYC fragments in our overexpression experiments might either be a result of an internal methionine start codon or an internal start codon in our MYC gene, or it could be a result of cleavage. So to figure that out, we mutated internal methionines in our overexpressed MYC. However, despite that, we still see this shorter MYC fragment being produced, indicating that this shorter fragment might be most likely be a result of proteolytic cleavage. So we wanted to ask, what are these fragments? How are these fragments made? And why are these fragments produced in the first place? So to begin to answer that question, we first asked, where does MYC cleavage occur? And to do that, we adapted this cleavage reporter that was first done in vitro, and we wanted to see whether we can do it in our cell reporters. And so we made use of HNUY, which is a protease-resistant protein from P. gingivalis, and we used it as a protease-resistant scaffold in the N and C terminus of a reporter construct. And here we're putting different regions of MYC in between these to probe whether they can be cleaved or not. And you can see that the N terminus has an HA tag, while the C terminus has a flat tag. So we put regions after MYC box 2A, or the region around MYC box 3A, and the region from MYC box 3B to MYC box 4 to probe whether this particular regions can be cleaved in our cells. And so when we're looking first at the N-terminal cleavage products of this, we can see that the region surrounding MYC box 3A, as well as the region after MYC box 3B, often produce N-terminal cleavage fragments. And I do want to give a shout out to their amazing undergrad, Yana, who has helped me in this part of the project. So yes, so we can see N-terminal cleavage fragments, but more satisfyingly, we can also see the corresponding C-terminal cleavage fragments using the flag antibody. More satisfyingly, we're able to as well pull down these cleavage fragments using HA IP as well as a flag IP, indicating that in cells, MYC seems to be cleaved in this region around MYC box 3A, as well as this region from MYC box 3B to MYC box 4. So to confirm whether this cleavage can occur in vitro and to probe as to where MYC is being cleaved in the cellular compartments, we then made use of this in vitro reporter, where again we're doing this in a test tube now, where we have the protease-resistant scaffold HNUY with a C-terminal flag tag, and at the N-terminus we put the region surrounding MYC box 3A and the region after MYC box 3B. And you can see here that we're able to in vitro purify from bacteria these reporter constructs pretty well and pretty cleanly. So we got cytoplasmic and nuclear fractions from U2OS cells, and I wanted to see whether they can cleave our reporter constructs over here. So you can see that the region around MYC box 3A, adding the cytoplasmic fraction of these U2OS cells does not lead to cleavage of this reporter. However, when we add the nuclear fractions of this, we often see this shorter cleavage fragments that are being produced when we add the nuclear fraction of U2OS cells with this MYC box 3A reporter. Now, looking more so at the region after MYC box 3B, you can appreciate that adding the cytoplasmic fraction leads to proteolytic cleavage of this reporter. However, looking at the nuclear fraction, this particular cleavage by the nuclear fraction is much more apparent and seems to be more stronger. So we wanted to ask, what is this nuclear protease that seems to be cleaving MYC at these particular regions, and how is MYC cleaved? And so the evidence for this actually comes from old data from a c-MYC homolog in C. elegans called MML1. So here we have this transgene of MML1 with a GFP and flag tag on the C terminus. And we put them into the wild type or a ZFP1 mutant cell. So in the wild type cells, dot 1.1 can interact with its binding partner, ZFP1. And so here the dot 1 complex is good to go and it's functioning. And so dot 1 is brought to the chromatin to its target genes. However, in the ZFP1 mutant, it is a C-terminally truncated ZFP1, and this association between dot 1.1 and ZFP1 is lost, and therefore dot 1.1 cannot be recruited into the chromatin. So this transgene should be around 150 kilodaltons. Now, when we put it into the ZFP1 mutant, where the dot 1 complex is compromised, we do get to see the full-length transgene being produced at around 150 kilodaltons. However, in the wild-type worms, we do not see the production of this full-length protein. Instead, we get the production of this around 60 kilodalton shorter fragment over here in the wild type that seems to be diminished under dot 1L compromised conditions, indicating that there seems to be this cleavage relationship between the smaller fragment as well as the full-length fragment, and that this cleavage seems to be facilitated by the dot 1 complex. So, we wanted to ask, how does dot 1 facilitate c-MYC cleavage? And this actually comes from insight from the transcription factor NRF1, which is activated by BCP as well as proteolytic cleavage. So what we know so far is that our results suggest that MYC cleavage is occurring in the nucleus based on their nuclear fraction experiments in vitro. And this is where dot 1L is functioning. And we've shown that dot 1L is functioning in the nucleus with MYC in relation to its extraction with BCP. And that dot 1L cooperates with BCP. We've also shown so far that in worms, the dot 1 complex facilitates MML1 cleavage, which again is a c-MYC homolog in worms. Now, it turns out there's this other transcription factor that is activated both by BCP and some proteolytic cleavage by a protease. And so here, and that is NRF1. So NRF1 is recognized by P97, or which is BCP. And BCP then leads to the extraction of NRF1 from the ER lumen onto the cytosol. This would then lead to the proteasomal degradation of this transcription factor. When the proteasome is inhibited or perturbed, however, BCP extracts NRF1 from the ER lumen, and this protease called DDI2 leads to the cleavage of this transcription factor, leading then to its translocation into the nucleus and leads to the expression of genes related to the proteasomal pathway. As an aside, DDI2 prefers ubiquitinated substrates. And so there could be, during our research, we thought that there could be then a DDI-like protease that favors ubiquitinated MYC as a substrate. So we wanted to ask, what is this mystery protease that cleaves MYC? Or also there's a possibility that maybe dot 1L itself might have proteolytic activity. So dot 1L doesn't really has not been shown to have any proteolytic activity at all. But despite that, we took the time to then look whether there is any homology of dot 1L with certain proteases, and more importantly, with certain DDI2 proteases. So we aligned the histone-methyltransferase domain of dot 1L with DDI proteases. And surprisingly, the histone-methyltransferase domain has pretty good homology with the catalytic site of DDI proteases that are related also to the HIV protease. And you can see here, especially in this boxed region, which is the region that dot 1L uses to interact with the S-adenosylmethionine, you can see that this catalytic aspartic residue of the DDI proteases aligns very nicely to this aspartic residue in dot 1L, which again is very important for the histone-methyltransferase activity of dot 1. Apart from that, smart homology also identified this C-terminus region that has similarity to a fold in serine proteases. So we wanted to ask whether dot 1L itself has proteolytic cleavage activity. And to do that, we purified the C-terminus of dot 1L that is highlighted here in the yellow, and we were able to obtain the N-terminus of dot 1L from commercially available vendors. And so we wanted to probe these in vitro reporters again, whether they can be cleaved by dot 1L. And you can appreciate here that the region surrounding MYC box 3A, when we either incubate it with the C-terminus of dot 1L or the N-terminus of dot-1L, we don't really see any cleavage products occurring. However, when we incubate the region after makebox 3B with the N-terminus of dot-1L, we see that cleavage products are being produced upon this incubation, and that this is dependent also on the concentration of dot-1L. Now, if we look back into our in vitro cleavage reporters with the nuclear extracts from U2OS cells, you can appreciate that the pattern of the cleavage products that are occurring from the region from makebox 3B to the basic helix region are similar in size and similar also to the patterning. So this gives us a little bit more confidence and evidence that potentially dot-1L is this protease in the nucleus that is cleaving MYC in cells as well. So we wanted to also see whether dot-1L can cleave full-length MYC. So we got commercially available recombinant MYC and also incubated it with either the C-terminus or the N-terminus of dot-1L. And you can appreciate here that incubating with the C-terminus, we don't really get any distinct bands compared to the control. However, incubating it with the N-terminus of dot-1L, it leads to the cleavage of MYC, and we can see the N-terminal cleavage product over here. When we added the nuclear fraction as well onto this full-length recombinant MYC, you can see here that the main cleavage product occurs with the nuclear fraction and also occurs at around the same size as a cleavage fraction with dot-1L. You can also see the corresponding C-terminal cleavage fraction fragments using the C-terminal antibody of MYC, and you can see here there's a weak main cleavage fragment of the C-terminus when we add the N-terminus of dot-1L with recombinant CMYC. And again, when we add the nuclear fraction, we're able to see that the main MYC cleavage product matches in size with the cleavage product obtained from dot-1L. So you might be asking now, what is the purpose of MYC cleavage? And honestly, we don't really know yet. But we think that this cleavage of MYC might fit really perfectly with the first part of my talk that I discussed today, with this degradation cycle of MYC. And so we propose basically that this cleavage might be involved in this cycling. So where MYC binds the promoter, leading to gene expression, and dot-1L then cleaves MYC, allowing then potentially for BCP to recognize MYC a little more efficiently, leading to its extraction from chromatin. This then would lead to the degradation of MYC from the chromatin, allowing this whole cycling to continue and leading to gene expression. So for concluding, I hope I've shown to you guys today that c-MYC turnover at promoters is important for MYC target gene expression, that dot-1L cooperates with BCP for the removal of MYC at the target promoters, and that the histone 3 lysine 7-9 methyltransferase dot-1L has this non-canonical proteolytic function that may aid in MYC turnover. And with that, I would like to thank everyone who has made this research possible. Everyone in Alla's lab, especially Alla, who has been an amazing mentor, who's been very, very patient, and has helped me grow from a scientist who was very unsure of himself to a scientist who's a little bit more sure of himself. I'd also like to thank all the research fellows and postdocs whose work I've built upon during my 6 years, especially Katya and Alexandra, who helped make the knockout cells that I've been working on, to Ainhoa and Ruben as well as Daphne from Valentina's lab. I would like to give a special shout out to our awesome undergraduate, Yana Nikoric, who has contributed a lot to this project, and very proud how she's grown as a scientist. I'm very happy to announce also that our 2 undergrads who are graduating this year are both going to PhD programs. Yana's going to Cornell, so congratulations to her. I'd also like to thank, of course, my committee who have guided me throughout these 6 years in Alla's lab, and I appreciate all of your feedback very, very deeply. And I also want to acknowledge some of our collaborators in the Emily lab as well as Kathy's lab as well. And of course, the Genome Science Institute, the Genetics and Genomics Graduate Program, as well as PIBS, and some of our funding sources, and some people who have helped obtain resources for us. So some of the more sappy parts, thank you to Alla and everyone in the lab, especially Thomas. I really appreciate all your mentorship on worms as well, and our brainstorming during lunchtime. You've been a really great friend to keep me company in Allah's lab. Special shout out to everyone on the 4th floor, especially Dimitri, who has seen me from basically a first-year graduate student up till now. I'd like to thank all of my friends I made in BU. I hope I've made some lifelong friendships with y'all. And you guys have really been a great support system for my time here in BU. To all my friends who have come, yes. To all my friends, that's supposed to be a surprise. Oh God. To all my friends who have come here, who are outside of BU, and I've made from college and from beyond. Thank you. I really appreciate all your help and support and your friendship. I love you all, and you've been such a great support system here in Boston. Shout out to my 2 roommates and friends, Kurti and Fossey, who have heard this talk now 3 times. To the cats, thank you. And of course, thank you to my family, who have been, again, my #1 support system throughout my whole life. Thank you to my parents, especially. I can't imagine the amount of sacrifices you've had to make for me to get here where I am today. To my partner, Levan, you know what you mean to me. I love you. Yeah, thank you. If you have any questions, I'll be happy to answer them.
We have to finish crying.
Oh, no.
Yeah, go ahead.
Have you tried to meter transfer dot one L to see if the degree system goes on?
So the question was, have we tried to mutate the histological domain of .1L to see whether this would affect the cleavage? So I have tried to create our own N-terminal of .1L with the histological mutated, but we've had some issues in purifying this product so far, so we haven't been able to really test clearly whether the mutation would affect proteolytic cleavage yet. But that's basically the next step, right? So like do that, as well as we can also attempt to then create histological mutant cells in our actual endogenous cell line to like see whether that's affecting cleavage as well. But yeah, so that's the next step. We haven't done it yet, but it's something we'll probably have to do.
It's a great work. So how do you think specific is .1L is for me, or are there any other substrates?
Yes, so right now we just focus on MYC, but we think that there might be other substrates that .1L might be either cleaving or helping remove, one of which might potentially be NRF2, which also shows up in RNA-seq and proteomics analysis. So NRF2 is kind of a family member of NRF1, which we know to be cleaved by BCP and the DDI2 protease. So we think there might be other transcription factors that might be affected by this as well, but we just haven't looked there yet.
Really beautiful talk.
Thank you.
You got through 60 slides, and it was all really clear. You only talked a little fast. So, since you have that up, why is it necessary to have a cycle? Like you often, simplistically, you might think the transcription factor goes on, it stays on the chromatin, and transcription proceeds, and then it comes off. Why do you have to have cycles?
Yeah, so I thought about this a little bit. One is that I think potentially these transcription factors can exist in many different complexes, and this cycling allows them for the complexes to then different complexes associated with the transcription factor to then bind to the promoter. Another thing I thought about recently is that there have been studies also looking at the dwell times of these transcription factors at the target promoters, and it seems that these factors can have very, very varied dwell times that can also affect the expression of the target genes. So I think there might be, it might not be a very static process, but this temporal dynamics is very much important, I think, for the expression of